I have the pleasure to announce or to introduce Mas Dojiri from the LA City Sanitation and Environment. He's going to handle the next panel, as Miguel did on the last. And Mas is the assistant director for LA City Sanitation, and he's also the chief scientist. We have had a partnership, as I said earlier, with LA City Sanitation for many, many years, and Mas has been like our rock partner, always working closely with us. We work on these events in past years, and he's been so critical to us bringing all of you together over these many years and when we've had these symposiums. And so the next panel, he will introduce the from source to stream panel, and he'll talk to you about how he plans to handle the speakers and all. But would you please welcome Mas Dojiri?
Hi, everybody. I trust that you can hear me. I have a booming voice, I think, that carries. Got me in trouble in elementary school and junior high school for quite a bit. But now it's a positive thing, so there. As Rodney Dangerfield would say, what a crowd, what a crowd. I mean, it warms my heart that you guys would want to attend this symposium and learn about the LA River and the LA River Watershed. So I think that's great. One of the advantages of attending these symposiums or symposia is not just learning about the LA River and the LA River Watershed or whatever technical sort of information that is presented, but so much of it, I realized when I walked through the doors after registration, was networking and seeing some friends that I quite honestly didn't expect to see here. Not because they don't belong or they shouldn't be here, because it just didn't click in my mind. And to see these colleagues who have become friends just warms my heart. And it's just another plus for attending here. One of my favorite TV shows, this is kind of off to the side now, but it's the Diners, Drivers, and Dives with Guy Fieri. And he had an episode on a bar in Annapolis, Maryland. And, that's one of my favorite towns anyway. But anyway, the bar had a sign right above the bar, and it said, "There are no strangers, only friends whom you have not yet met." And that's how I feel about this symposium. I see all these friends, but then I see all these attendees and part of the audience, and you, as far as I'm concerned, are friends whom I haven't met yet. Okay, so that's my opening statement. I want to say good morning and welcome to panel 2. Steve mentioned it is From Source to Streams, Scientific Perspectives on the LA River Watershed. I'm going to keep my introduction short and my comments really short so that we have more time at the end for Q&A. The panelists should be in the first 2 rows. I will have them, I will follow Miguel's lead and have them sit there so that these lights aren't in their eyes like they are in mine right now. And they'll come up and make the presentation. They'll go back and sit down. But during the Q&A, they'll be up here, okay? This panel will focus on the science being conducted in the watershed to better understand stream conditions, biodiversity, water quality, as well as our efforts to improve the overall watershed health. The first presentation, the Los Angeles River Watershed Monitoring Program, will be given. Should I advance it? Yeah. Will be given by Dr. Yareli Sanchez, Senior Scientist with the Council for Watershed Health. Yareli?
Hi, good morning, everybody. Good seeing you all 5 years later. We first met in 2018, and now I have the pleasure of reporting on our stream assessments that we've been doing over 15 years. And this data really does inform our understanding of the state of the watershed from an ecological and a little bit from a public health perspective. And so I'll be reporting to you today about some of those comprehensive stream assessments. And so you've heard a little bit about the program earlier today in Barbara's remarks and in Moss's remarks, but a little bit more about the LA River Watershed Monitoring Program, which we call LARWMP for short. It's a watershed-wide comprehensive monitoring program that began in 2008, and we complement and coordinate with regional and state entities, so our data is comparable with some of the other stream assessments that are happening in the state. The objectives of the program are to provide a complete understanding of watershed and how it is changing, and to create a program that is responsive to the priorities of managers, river stakeholders, and the public. All the activities under the LARWMP program are guided by these 5 questions. I'll be reviewing the main results of these five questions that guide our program. So 1, what is the condition of streams in the watershed? Are conditions at areas of unique interest getting better or worse? Are receiving waters near discharge meeting water quality objectives? Is it safe to recreate in the streams and in the river? And are locally caught fish safe to eat? And so before I get into some of the results, I do want to note how we collect samples, and for many of the random sampling that we do to get a good picture of what's happening watershed-wide, we do random sampling in these 3 subregions. So the natural subregion, which encompasses the Angeles National Forest, the Verdugos. We do sampling in the urban tributaries, and in the effluent region, that main channel of the river that's dominated by those POTW flows, those public work flows. And so I'm going to move quickly through our program here because there is so much data to present. If anyone is a data wonk, we have lots and lots of data all publicly available. I am not presenting all of it here today, and we'll just be sort of highlighting the main story from the assessments analysis that we've been doing. So question number 1, what is the condition of streams in the watershed? So to assess this, we look at biological indicators. I have them pictured here. We're looking at algal communities and benthic macroinvertebrate communities. These communities integrate stress over the long term, and when we measure them, we sort of bring the different measurements into a multi-metric score called the California Stream Condition Index for benthic macroinvertebrate and the Algal Stream Condition Index for algae. And through those measurements, we get a better understanding of how healthy or intact streams are or how impacted they are. And so from these biological indicators, we found that between 54 to 72% of sites in the watershed are in some sort of altered condition. And you'll see, we have the algal data to the left there, the ASCII and the CSCI data based on benthic macroinvertebrates to the right. And you see that, that we have the different subregions. The natural area tends to perform better, as you might expect, than those effluent and urban regions. And then we have a lot more impacted sites in effluent and urban regions than we do anywhere else. And the majority of sites are impacted in some way based on these biological indicators. In 2018, I came up here and I said, at all of our sites, we see trash, and trash is a problem. We should really get a better understanding of the extent of the trash issue. And so we have done that beginning in 2018. We collaborated with the BITE program and started doing these visual trash assessments, and we continue to collect that data year after year. And so what we have found, which is probably not very surprising to the folks that care about our waterways, is that the greatest abundance of trash is plastic trash. And we also see large abundances of metal, biodegradable, and fabric and cloth. But overall, trash is the most abundant trash item at our sites. And then looking at from a percent stream kilometers assessed perspective, we find that there is some sort of plastic at 48% of our sites, and there is some sort of trash item at 61% of the stream kilometers that we have assessed. And I have some picture of our teams doing trash assessments and what our sites look like. And so moving into the other question, are conditions at areas of unique interest getting better or worse? We have sites that are important because they're wetland ecosystems and riparian ecosystems, and we know those ecosystems are few and threatened. And many of them are present in the urbanized regions of our watershed, and so we've been following them for 15 years, and we've been doing riparian habitat condition assessments, and so I'm showing you that data here. And what we're finding is, we see that same pattern where sites in the upper watershed are healthier than our lower watershed sites. But the good news is we've been doing trend analysis to see how things are changing, and we're finding that our riparian habitat is stable. It's not getting worse over time. And I have a picture of some of the sites here where we have that are impacted and have CRAM scores, California Rapid Assessment Method scores, that show that they're impacted. So that includes the Sepulveda Basin, the Glendale Narrows, and Eaton Canyon. Next, we look at receiving waters, and we, take samples above receiving waters and below those discharges, and we look at a slew of parameters. Here I'm presenting E. coli and ammonia and nitrates. And we find that there's elevated E. coli at some public works plants that are higher than the upstream location. So we can see that for the Burbank plant and I believe the Glendale plant. So in yellow we have the downstream concentrations, and so they're higher than the blue bars there. And then in terms of nutrients, we find that nutrients downstream of those discharges tend to be elevated, but they're meeting water quality objectives for nitrate. And so that water quality objective there is signified by the red line. Then the latter two questions are most focused on public health. So as part of these questions, we look to understand whether it's safe to recreate in the streams and the swimming holes in the watershed. As you can see, they're pretty popular with the public during summertime. And we go out there about once a week during the summer and take E. coli samples and compare them to water quality objectives to see whether they're safe and it might cause gastrointestinal illness for any of the folks in the water. That data is publicly available and reported on LA Sanitation's website. And what we found over the years, and as we're doing sort of the 15-year assessment, is that there are sites that are more frequently exceeding the others, and those tend to be Hansen Dam and the Bull Creek sites. And so you'll see those pie charts are mostly, a bigger proportion of them is red, so they have a higher number of exceedances. And then finally, we're looking at our locally caught fish safe to eat. And so we take samples in the river and at lake sites in the watershed because we have a lot of anglers that are out there consuming the fish that they do capture. And so we look at some of the contaminants that are bioaccumulating and biomagnifying. We collect mercury, PCBs, DDTs, and selenium. I'm not presenting DDT or selenium because they tend not to be elevated in any of our samples. But for mercury and PCBs, we see that it's generally safe to consume the fish in our watershed, sort of depending on where you're at. And the fish species that you're consuming, some of the sizes of the fish that you're consuming will have to vary. And so for the largemouth bass, for example, at Leg Lake, Belvedere Lake, and Echo Park Lake, the amount of mercury in those samples mean that you should only consume one serving of fish fillet from those sites. And then in Leg Lake, one serving of white catfish, and everywhere else you can consume between 2 to 3 servings of those fish based on our sampling efforts. And so I'll leave you with the recommendations for the LARWAMP program. We know our watershed is changing and rapidly changing and will continue to change. You'll hear more about the efforts that are taking place in the watershed from the presenters that will follow me. And so some of those include, the more wide-scale implementation of stormwater best management practices, reduced flows, and then there's also habitat enhancement and restoration that's happening in the watershed. And so the monitoring is really important to understand how those things are working, the impact to the health of our streams, and how we should be managing, particularly under a rapidly changing climate. And so recommendations for the LAR1 program is that we continue to collaborate and coordinate with the folks that care about the health of our waterways, and with the managers that are making the decisions that are impacting how that land is being managed. Thank you so much. That's all I have, and I'll answer questions at the end.
Thank you so much, Yareli. As mentioned before, prior to 2008, we conducted site-specific monitoring right at the point of effluent discharge, a little bit above or upstream of the discharge and a little bit downstream. Certainly a more regional monitoring program is much more informative for the LA River than the site-specific monitoring that we used to do, especially a program that is focused on those 5 management questions. So thank you very much, Yareli, for the update. The second presentation is titled Constituents of Emerging Concern, CECs in the Los Angeles River, and will be presented by Dr. Charles Wong, head of the Chemistry Department at the Southern California Coastal Water Research Project, also known as SCORP. Charles?
Okay, so good morning. Thanks for coming. I'm glad to be here. So I want to talk about CECs in the LA River. So I want to talk basically about 3 things. First of all, what are CECs and why do we care? And then basically dive into what is the issue in the LA River, where I want to talk about generalities, what is the state of California, which of course is the LA River is in, doing about CECs, and then also a little bit about what we know about CECs specifically in the LA River. So I want to talk about each of these in turn. So what are CECs, constituents of emerging concern? They're chemical contaminants. But unlike what Yareli was talking about, things like PCBs and PCBs and mercury and such, where we know a lot about them and there are regulatory levels for them, and we have a good sense of what we want to do. For these chemicals, they generally don't have a well-defined regulatory standard, and the reason why they don't have a well-defined regulatory standard is because there's a lot that's generally not known about them. So for example, it may not be known what they do to things or what they don't do to things in the environment, organisms living there. We don't necessarily know how they behave. The classic paradigm in toxicology is that the dose makes the poison. So if you don't know what controls the dose, you don't necessarily know how bad it is. We don't necessarily know where they're coming from, and in many cases, we don't even know how to measure them. These could be brand new chemicals. So for example, replacement flame retardants in things like, the cushions and the seats that you're sitting in. We've heard a lot about PFAS in the news. Many of those chemicals have been known for a while. Some of them are brand new. They're just being discovered. Or they could be chemicals that we've been used for a long time, things like pharmaceuticals. But for example, the capability of being able to measure them only started about 25 years ago. Clearly, people were taking pharmaceuticals a lot longer than 25 years ago. So another paradigm of this is that typically when we think about chemical contamination, we think about smokestacks or industrial activities, but these are also coming from day-to-day use, household use. So they come from all sorts of places. So what makes them especially of concern is that there's ever-increasing numbers. So this is a plot of all the chemicals that have been registered with the American Chemical Society's Chemical Abstract Service. They hit 100 million chemicals in existence as of 2015, which was 8 years ago. So you can imagine the number is more than 100 million. And again, for a lot of them, we don't know what they do in the environment. We don't even know what's out there. So how do you deal with them? So I just hit, there we go. So for obvious reasons, we're not monitoring 100 million plus chemicals. I mean, that's just not feasible. So, and monitoring work is very expensive. So it's, how do we deal with trying to figure out which ones to monitor? We could playing whack-a-mole all day, but what we want to do is hit the target, which is to ensure environmental quality, good water quality. So we need paradigms to understand what would make good water quality with regards to dealing with these new chemicals and these emerging chemicals. So that's where I want to seg into generalities in terms of what the state of California is doing about CECs. And the state of California recognized that there's a huge potential problem with these CECs. Which ones are worth monitoring? Which ones are things where that aren't going to be a problem, and are things that we can devote efforts into other things that are more pressing? So what the state of California did was, about a decade ago, it formed an advisory panel, a scientific advisory panel, and it charged the panel with a strategy for figuring out how do we deal with this, how do we prioritize what to look for and what to deal with, and what to spend our resources on. So these were experts, worldwide experts, in a number of different interdisciplinary fields, ranging from chemistry to toxicology to risk assessment to engineering. And what they did, they came out with a report, it's publicly available, was they developed a risk assessment framework to figure out which of these chemicals we should focus our limited resources on. And this was based on risk assessment framework that emphasized chemical occurrence and chemical toxicity. In other words, how much is out there, and is it too much? Because just because it's out there doesn't necessarily mean it's a problem. So that also, those answers also figure out what to do about it. Should there be monitoring? Should there be compliance work? Should there be special studies to get a better sense of what levels are, and are they getting better or worse? So this framework has been adopted by the state. So the State Water Board, the regional water boards, and many other public entities that are dealing with CECs has incorporated this as part of the strategy towards managing CECs within their own areas. So we can use it, as I'll talk about later, we can use this in terms of case studies, especially in the Los Angeles River. Now, one of the recommendations this panel did, which the state also adopted, was the fact that this should be reassessed every so often, because these are new chemicals and there's new data being produced, new information, and also advances in technology and in the state of the art of the science, so that you can measure these chemicals and understand more about their toxicity and their behavior. So in order to stay up to date, what the state did was reconvene a new panel in 2020 to update the strategy that the original panel had. Using the data that became available, I'll show you an example of that in the next slide, and incorporating all the advances happened. So this panel just wrapped up. I chaired it, and they produced a report that just came out. Again, it's also publicly available, and basically what it did was it said that the framework that was originally there was sound. There are other ways of doing this, but the framework that the state adopted, that the original panel suggested, did make sense, and it also provided guidance on basically how to improve the data. So, for example, what we have plotted here is one of the recommendations that the State Water Board promulgated as a result of the original panel, which is collect and compile monitoring data. The original panel had real problems with because they didn't have all that much data to work with. So in the interim, from basically 2010 onwards, these dots represent places where there has been monitoring work at least once for several hundred different types of CECs, and that has been compiled within a central database. So if we looked at, if I showed you the map basically before the first panel, there would be very few, if any, dots on it. So what I want you to get from this is not, oh my God, there's dots everywhere. There are dots everywhere, but this represents the extensive amount of monitoring that's been done by various entities, such as POTWs, various agencies, academic studies, so on and so forth, that have gone into CEC monitoring and produced the data to be able to say something that's useful with regards to how to monitor these CECs, and that's what the panel did in terms of dealing with this. So now I want to talk a little bit about the LA River. So there's a lot of information on there, so I'm just going to put some highlights that my own agency has done, where we've completed a couple of studies. Again, these are available in the scientific peer-reviewed literature on understanding what is the level of CECs at various points in the LA River, and is this a problem? This is using things like sediments, measuring things in water, measuring things in effluent, and also putting caged fish into the river and studying basically how the fish change in terms of, are their livers getting impacted, and also things like, enzyme and hormone and genetic changes compared to fish, control fish that were never exposed. So there were chemical analysis, and there were various analyses involving these fish. So this is an example of some of the chemical data. So what we have here are a number of different CECs, pharmaceuticals, pesticides, other things. I want to focus on the two heat maps on the two right ones. So the one in the third column is basically in Sepulveda Basin above Bull Creek. And you can see that there are some hits that are basically from things like urban runoff. And the one on the far right is basically downstream of the wastewater treatment plants that discharge into the LA River. For obvious reasons, there's a lot more hits. Now, these treatment plants are very advanced. They do an excellent job in getting rid of contaminants and pollutants. However, no wastewater treatment will get rid of everything, 100% everything. So this represents what little gets through. And the good news is that none of these chemicals exceed the monitoring thresholds based on what I told you the panels came up with. Now, in terms of effects, this is what we see in the caged fish. So we see concentrations of a couple of different estrogens. And again, look at the two rightmost plots in both of these plots. They're basically no different than the controls. What it's saying is that the differences in the estrogen levels in these caged fish, these are male fathead minnows, they're no different than fish that were never exposed to the water in the first place. If you look a little bit more, we look a little bit more closely. These are levels of vitellogenin. So what vitellogenin is, it's a egg yolk protein, and these are male fish. Male fish should have nothing to do with egg yolks proteins. They shouldn't be developing them at all for obvious reasons. However, if they're exposed to endocrine disruptors, they can. And what we note here is that the levels of these proteins in these fish are no different than the control. So what this is telling us is that while CECs can be a problem, there are strategies to deal with this statewide, and even though CECs are present in the LA River, the levels of the compounds that we know about don't appear to be causing problems in terms of things like feminizing the fish, and that continuing monitoring is something that we want to continue to do to ensure good water quality in the LA River and its watershed. Anyway, that's all I've got, and thank you for your attention. I'll be happy to entertain questions. Agreed. Thanks, Charles.
Thank you so much, Charles. I read somewhere, and I don't remember where, that chemists have synthesized over 100,000 new chemicals. Obviously, that's a lot less than the 100 million that you mentioned, Charles. So that article was like a few magnitudes off. But it also stated that chemists are synthesizing 1,000 to 2,000 new chemicals every year. So studying contaminants of emerging concern is certainly a daunting task and certainly relevant and very timely. So thanks for tackling it, Charles. I would not want to tackle that particular topic, and thanks for your presentation. Next up is Dr. Rachel Meyer, Assistant Adjunct Professor from UC Santa Cruz, and she will present on environmental or eDNA in the Los Angeles River. Rachel?
Hi, everybody. I've been waiting 4 years to be able to talk with all of you about this project. I'm sure we all feel that way. So thank you for having me here today. Who here I get a show of hands, who here has heard of eDNA, or environmental DNA? All right, that's fantastic. So quick review, eDNA is what we shed into the environment. It can be free, it can be inside cells or sloughs of tissue. You can pick it up on this microphone. All of us who are speaking into it have DNA on this microphone. You can pick it up in a scoop of water or soil or from dust and air. And it's really an exciting tool now to be able to do biomonitoring for all sorts of things, not just taxonomy, like what species are there, but maybe even in the future, what are the functions? At UC Santa Cruz, I'm part of also the paleogenomics lab, where we go back into the past. So we have eDNA projects that go back to 33,000 years before the arrival of humans in California. And we're trying to apply these methods, looking at ancient and degraded eDNA to try to help the field move forward. So I'm not part of a team that does conservation work to save anything. We really just want these methods to be better so that you have the tools to use this biodiversity information to do your work. So I'm part of CALeDNA also. I've been directing that program since it started in 2017. And we collect environmental samples all over California, and now actually all over the world. Just small tubes of environmental samples that go in a minus 80-degree freezer, so that in 100 years you can come back and say, what was the biodiversity at that point in space and time? And we build web tools to help people understand the data. We also have a service lab where we generate the eDNA data for you. We largely use a technique called meta-barcoding that I'm not going to get into at all. And we have many grassroots projects all over the state that we try to help people integrate together so we can all learn some lessons and help each other. One of the projects is on the LA River. This was started by some generous funding from Metabolic Studio. And many of you in this room were a huge part of this project that made it possible for us to gain access to sites, to be able to collect samples. Many of you shared your own data sets so that we could understand how the river varies and try to see how the eDNA would plug in. And together at a workshop, many of you were there, where we selected the sites to study on the river. And we ended up collecting environmental DNA samples with the goal that we would connect educators and scientists, make an open data platform with the results so that we can enable you to monitor biodiversity. From the headwaters to the ocean, looking at differences that people were interested in, like soft bottom and hard bottom, what the river width is, how that varies with biodiversity. And we did that in all three of the major waves of COVID, by coincidence. And so we have all these environmental samples that apparently are going to be useful when the Olympics are here. But we're continuing to do some monitoring. So we've actually got 10 time points from 2022 and 2023. There's a BioBlitz this weekend if you want to come help us sample. I know Levi somewhere in the room really needs some help. He's in the back. So all of this is now online. We have about 500 samples that we've processed for 6 different meta-barcodes that cover the entire tree of life, well, imperfectly. Originally, we had put them on our CALeDNA page where you can view results, but it wasn't that interactive space that we really needed. And we're not the only people doing eDNA monitoring on the LA River. So we really needed a space that scientists, practitioners, students, all sorts of different members of the community can interact with the data. And we know that eDNA is just sort of in its infancy for a tool, that there's going to be a lot of improvement in sequence data that we compare our eDNA data to be able to make discoveries. And so we want to be able to reanalyze those data sets. We want to be able to do that in a quality way as the field moves forward, to be able to continuously improve. And that there's a lot of uncertainty with eDNA. So when I started working on CALeDNA, we would give people a species list of like, here's what we found in your reserve. And they would look at it and say, oh, I don't recognize a couple of those things. Throw it away. Right? There's a lot of distrust in eDNA, and many of you may feel that way still. And we think that uncertainty is also an opportunity to start to play with that uncertainty, figure out where do we fill gaps, how do we improve this as a tool. For example, you might want less uncertainty in the taxonomic accuracy if you're making a species list. But you might want to allow more uncertainty so that you reduce bias and being over-conservative if you're trying to do alpha or beta diversity assessment for your community ecology. So this is for you, all right? That Oprah Winfrey, you get Vaseline, you all got a giant eDNA data set, okay? Please go on your phones now to the eDNAexplorer.org, eDNAexplorer.org, no the, I don't know why I said that. And you can click on the LA River, and it's still a little slow, right? We just launched this, like soft launched last week, so this is our medium launch. We're not fully officially launched, but please check it out. You can explore You can explore and learn about the LA River's environmental DNA and many other projects already that are up there. You can analyze your own data. You can share your project with your contributors and use this as a project management tool. And hopefully make some discoveries about how biodiversity works. So here are some examples of projects that are on the eDNA Explorer. We also have some wildfire projects looking at recovery and how other watersheds in California are reacting to water primrose as it invades. And many of your names are up here. This probably deserves to be doubled. So we're still building this out. You might get an email when we add you. So this is the project page for the LA River project from 2020 to 2021. The pilot data is also online. And we present to you an organism list that you can filter in many different ways. And we really focused on making this site accessible and really beautiful. So we worked with this amazing team that's our co-leads on this project, the eDNA Explorer project, called Sliced Bread Design. They are user-focused, so they actually did many interviews with people to figure out what you needed, and then this site is completely designed around you. So there's the frequency of samples that have those organisms. Here's a flip through of some of the vertebrates: possums, perch, raccoons, green sunfish we just saw in the last talk, rats. Any surprises here? Cats. Okay. You can also do some basic sort of teaser analyses of alpha and beta diversity, looking at how different the sites are from each other, or looking at other metadata that we automatically give you, like human impact, light pollution, greenness, and we would love to include more social, sort of social science statistics to help us understand how more of our world relates to biodiversity and shapes it. So you can see, oh, there's a significant relationship between the vertebrates the vertebrate communities and the amount of artificial light. You can see how each organism varies in its occurrence over time and over space over your sites. This is the over time example. And you can download lists of species that are from other really complementary observations like iNaturalist and museum collections through GBIF automatically on our site. The machinery that holds this all together is called Tronco, and it means tree in Chamorro. It was developed by Lenore Pipes and Rasmus Nielsen at UC Berkeley. This is like a stealth phylogenetic super awesome analysis platform. It's fast. It takes me a month to analyze eDNA data. Now it takes me a day, and it's just as good, if not better. So you can upload your raw data, your FASTQ files, onto the site and run them through Tronco and compare that to what you're currently using with brand new reference databases that we will be annually updating. What I think is really cool about this is you get more species that you can identify, and then you see more patterns. So here, human impact is significant with Tronco when our old software, Anacapa, could not identify that global human modification or human impact was significantly varying the species richness on the LA River. So I'd love to talk with you all more about that if you're interested. Please check out our website, give us feedback, help us build some community. We're going to have another celebration of this data set in December. I'd like to thank Oceankind, who really saw the different collaborators, this amazing list of people working on this LA River project. as an opportunity to create to support making a tool that would help eDNA get into your hands. So thank you, Oceankind. Thank you all. I'm going to shut up now, and I'll take some questions later. Thanks.
Thanks, Rachel. And when you emailed me the eDNA Explorer, I did look at it, and it's awesome. It's really, so I encourage the audience to go ahead and take a look at it, especially for the LA River. You know, on a personal note, my first job with the city of Los Angeles was a first-level marine biologist. It's called a Water Biologist 1 position. And one of the tasks that I was assigned was to identify species in Santa Monica Bay and the Los Angeles Harbor to assess the effects of the effluent discharge on the organisms inhabiting these waters. You know, the traditional taxonomic methods that I used was incredibly cumbersome, tedious, very time-consuming. So for you know, the advancement of eDNA and using it to identify the organisms down to the species level is a huge step forward, and certainly close to my heart, so thank you very much for your work on that. Next up is Dr. Christine Taniguchi-Quan, a senior scientist also for from the Southern California Coastal Water Research Project, or SCWRP, who will present on the Los Angeles River Flow Project, development of decision support tools for flows in the Los Angeles River. Kris?
Great, thanks so much for the introduction, Moss. I'm Kris Taniguchi-Quan, and I also wanted to thank LB for sort of introducing and giving a little teaser to our LA Rivers Flows project. So I'm going to give you a brief overview and a little touch on some of the technical tools that we developed through this effort. Before I begin, I also want to acknowledge the technical team from SCWRP, Colorado School of Mines, and the Council for Watershed Health, who all contributed to this study as well. So in the LA River, during the dry season, during the summertime, in-stream flow is primarily from storm drains, as you see on the picture on your left, from the watershed, and also from treated wastewater or effluent discharged from the wastewater treatment plants in LA, Glendale, and Burbank. Now, as these municipalities recycle more water, they ultimately are going to be discharging less into the river. And so reductions of these discharges could lead to consequences to fish and wildlife and recreational uses that really rely on stream flow in the river during important times of the year. And so through the 1211 wastewater change petitions, wastewater dischargers are required to demonstrate that any change to their discharges aren't going to unreasonably harm aquatic life and recreational beneficial uses of these rivers. And so these 1211 wastewater change petitions are subject to State Water Board approval. And so that's where this project comes in. We wanted to develop a tool set or a toolkit that can help evaluate the potential consequences on beneficial uses from reducing wastewater discharges and also from increases in stormwater capture throughout the entire watershed. And so these tools will allow us to evaluate potential wastewater change petitions, stormwater management programs, and also in the long-term restoration efforts that are happening along the main stem of the LA River. And so what we did in this project is we developed a very detailed watershed model that simulates rainfall, runoff, and water quality from the entire landscape of the LA River watershed. And we had a special focus on the main stem of the LA River where these 3 wastewater treatment plants discharge, and also on the 2 tributaries of Compton Creek and portions of Rio Hondo, as you can see in pink here. And I just wanted to remind everyone that our main focus is really on understanding the effects of reductions in effluent discharge on the main stem of the LA River within the banks of the LA River, as you can see the flood control banks highlighted in red right in here. And so our three main goals, the first one is to really understand and evaluate the relationship between stream flow and changes in stream flow and beneficial uses in the LA River. And when I say beneficial uses, I'm referring to aquatic life like important riparian habitat, wading shorebird habitats, and also recreational uses like kayaking and fishing that rely on flows in the river. And once we understand this relationship, we can develop tools that allow managers to evaluate the potential effects of stormwater management, stormwater capture, diversions from storm drains, and changes in wastewater discharges that may impact in-river flows on the LA River. And a big part of this project was really engaging with the local community and stakeholders in the LA River Watershed as well. And we received really valuable feedback and input throughout the entire course of this study. And so before I jump into some of the technical tools that we developed, I wanted to just hit on some of our main outcomes of the project. We developed a set of tools that can be used to identify flow management targets. So what types of stream flow, how much stream flow during what time of the year should be managed in what portions along the LA River. And we also developed tools where we can evaluate the potential effects of changing river flows from management scenarios, from wastewater treatment plants and from stormwater management programs. And these tools are developed to be highly flexible and transferable. It could be utilized in other similar watersheds and publicly available for any of you to use and look at. I provided the link right here at the bottom. And then lastly, there's been broad consensus and agreement from our stakeholders on the application and the utility of these tools. And so as I mentioned, we developed this detailed watershed model that simulates hydrology, so stream flow, water quality, hydraulics like depth, velocity, and scour, stream temperature, and a stormwater management model that can help us simulate future management scenarios. So with these linked watershed model, we can now evaluate how environmental flow or stream flow characteristics may change under various management scenarios of wastewater reuse, stormwater capture, and diversions from the storm drains. But what we really care about is not only how do stream flow characteristics change, but how does that impact recreational uses and aquatic life. And so we also conducted a recreational use survey that Yareli Sanchez led for that aspect of the study, where she did targeted interviews and workshops with recreational experts to try to understand the flow needs to support different recreational uses along the LA River. And we've also developed species occurrence models, and that was done by Katie Irving, who's here from SCWRP, that allows us to understand what's the probability of supporting different species based on changes in stream flow. So that's the link between stream flow changes and the probability of supporting species. Now, with all of this detailed information, we ultimately developed what we call a sensitivity curve that allows us to look at different management trade-offs. And I'm going to go through an example with you at the end. And so, as I mentioned, there's multiple different beneficial uses that have been designated along the main stem of the LA River. These are the aquatic life uses that have been designated, different habitat types that need to be supported. So for each of these habitats, we identified end member species. So species that are indicators for that habitat type, but also represent kind of a wet and a drier end of flow needs for each of these habitats. And so for every single habitat and end member species, we developed a species occurrence model where we can relate changes in stream flow to species occurrence. I do want to note that the two rows in yellow, so cold water habitat and migration habitat, are not currently designated in the LA River, but there is a desire to evaluate if there's a potential to restore conditions to be more conducive for these ecological endpoints in the future as we think about restoration efforts. And so just to give you a quick little idea of what our relationships look like, we have flow on the x-axis down here. So as you change flow, you change your probability of a species occurring. And so we developed these tools, and we can use this curve to identify flow management targets based on a high probability and maybe a medium probability. So you could see for every species, we can kind of bracket an optimal range of flows that are supportive for different habitat types. And so ultimately, we can visualize our flow management targets or the optimal flows for different ecological endpoints. This is one example from Glendale Narrows, which shows the range in flows to support willow species and also typha. And there might be kind of an optimal flow range to support multiple ecological endpoints. And so these targets can be developed for anywhere along the LA River main stem and across different beneficial uses as well. And then lastly, we developed sensitivity curves to assess the effects of wastewater reuse. And so what you see here is an example curve that relates the dry season effluent discharge, so what the wastewater treatment plants are discharging into the river, and how that might impact the in-river, in-stream flows. So under baseline conditions where we have very minimal wastewater recycling, you can see this dotted line, and then as you recycle more water, you see there's a drop in in-stream flows during the dry season. And so we can overlay this curve with the flow management targets to evaluate trade-offs between reducing flows and the flows needed to support different endpoints. So this is one example where there may not be a lot of flexibility in reducing flows for willow and typha, but if you wanted to support willow only in this reach, you may have a little bit more flexibility in wastewater recycling. And then lastly, I don't have a lot of time to go into this, but we did also want to look at how these different management scenarios might impact water quality, because it's not just about the amount of water, but it's also the quality that matters as well. And we also looked at a restoration analysis to see if are there changes to the channel configuration within the flood control banks that can help us make more out of the water that we do have, or the reduced amounts of water that we will have in the future. And with that, I just wanted to thank all of our stakeholder community for providing input from the onset of our project. We had a year-long scoping project, from all the technical advisory meetings we've had to the recreational use surveys as well. And the stakeholder community really provided valuable feedback that strengthened the project as a whole. And with that, I also wanted to thank our main funding agencies and participating entities, and also you can see here all of my technical team members that helped make this project possible. Thank you.
Whose water bottle is this? Oh. Thank you, Kris. You actually mentioned all the post-presentation comments I was going to say, so I'm just going to cut to the chase and thank you for providing this model that helps both regulators and the regulated community to balance these two potentially competing environmental issues, water recycling and the attainment of beneficial uses. So thank you very much for that. It's a useful tool. We've already actually used it already for our 1211 change petition. So thank you. The next speaker is Maggie Gardner, Science and Policy Manager at the LA Waterkeeper, who will present her talk on community science at the LA River, water quality, and PFAS.
Hi, I'm Maggie Gardner. I'm the Science and Policy Manager at LA Waterkeeper. In my role, I help lead our community science program. The program itself is called River Assessment Field Team, shortened to RAFT. In our program, we bring folks from around the area to the LA River to learn more about the river, its history, the challenges it faces today, and our vision for what it could look like in the future. as part of the program, we integrate water testing to give participants more of a hands-on experience and to have conversations about water quality and how water quality impacts the conditions on the river. Oh, sorry. So for the water quality portion of our program, we use a variety of tests. For now, PFAS is not on here because it gets its own slide later. You can see them up on the slide in the top left-hand corner. You have dissolved oxygen, you have pH here, turbidity, coliform bacteria, and temperature. For the dissolved oxygen, this is something where participants will fill that with water from the river, and we put some tablets in it, and they have to shake it a lot to get a color change, which they'll match to this chart here. Similarly, for pH, they will also use a tablet in that and compare the color. For turbidity, they fill that cup there with river water and compare how easy it is to see the checkerboard sticker to a chart here. Coliform bacteria is one that is a 48-hour test, so it's not one we end up being able to show the results to them while they're there, but they just fill it up and the enclosed tablet does the work. And then one lucky participant gets to stick the thermometer in the water. So you've heard from several scientists today about their research, and you'll continue to hear more on this panel. I want to stress that our data from these water quality sampling kits in our community science program is very different from the data that's being produced from these other scientists. The purpose of our data is to engage participants around the LA River. We want them to gain a love of science and the LA River. It's not to collect high quality data. You saw on the last slide what our tests look like, trying to match the color of pH in that tube to a card is not going to be nearly as accurate as the methods and the lab equipment that these organizations have been using. That said, we had done a previous iteration of this program that was much more scientifically rigorous, but we found that it had a really high bar to entry. When we designed this iteration of our program, we were focused on making it accessible to anyone and everyone, no matter how much science you had under your belt. In this setup, the conversations that we're having with community members that are on the LA River is far more important than the quality of data that they're collecting. So the types of discussions that we're having are, why are each of these parameters important? What happens if one of the parameters is too high? What if the water's too hot or the water is too acidic? What if the dissolved oxygen is not high enough or the turbidity is too high? What do these have an impact on? What can prevent species from thriving or even surviving in the LA River? Because of this setup, we're also able to bring in summaries of research that is going on the LA River. So you've heard a little bit about the flows study. We often bring that in. We talk about the fish passage and habitat study. And I have some more studies I can now bring in at our next raft event. Now, simultaneously to this, we are also taking some PFAS samples. So Charles mentioned a little bit about this in the contaminants of emerging concern, but PFAS stands for per and polyfluoroalkyl substances. It's a class of chemicals. It's not a single chemical. And they're often called forever chemicals. They're a group of synthetic chemicals. They don't tend to break down in the environment, and they bioaccumulate, so they can build up over time in the environment, wildlife, and people. Some of the common uses that you might be familiar with are nonstick cookware, water-resistant clothing, and they've been on the market since at least the 1950s. Last year, LA Waterkeeper participated in a nationwide effort by the Waterkeeper Alliance to have each of their member entities take 2 water samples over the summer to get a snapshot of PFAS present across the United States. 113 chapters took place, and 83% of the samples that were tested across the U.S. tested positive for at least 1 PFAS compound. Now, after being introduced to the test during this process and seeing how prevalent PFAS was across the U.S. and in our own samples, we decided to incorporate it into our community science program. This is the one test that we do not do on site, and we actually end up sending to a lab. The test itself is pretty simple. We order sample kits from the lab we work with. The sample kit is an open cup that sits on a blue disc, and that blue disc houses a filter. The water in the sample, once you fill the cup up, will filter through that disc, and once all the water has drained and the filter is dry, I just box that up and send it to our lab for analysis. Unlike the rest of our water quality tests, this is one that we do end up placing stock in the results since we are having a lab conduct the analysis. Thus far, every single one of our 10 PFAS tests we have taken at various spots along the LA River have come back positive for PFAS compounds. While participants don't have access to the immediate results when they take it in the field, we do have conversations around emerging contaminants, how they get in our waterways, and our previous results, which so far have been pretty unanimous that they are present. Many of our participants have some familiarity with the term PFAS, but they don't necessarily know or understand what it is or what it can mean. So we tend to have a little bit more of an informational portion of our program and discuss a little bit about how that can impact our water and what it means moving forward. We also talk about what LA Waterkeeper does with this information, as I'm sure you might be wondering too. Right now, we're collecting this data to get an idea of what's happening in the LA River and in our surface waters. This isn't part of a study, and our only intention with the data right now is to use it internally to help guide some of our own decisions on how we engage on emerging contaminants. PFAS is an emerging contaminant, so there are efforts at the state and local levels to both research and make regulations around it. By understanding its prevalence in the LA River, that informs how our organization engages in these efforts and some of the ways that we choose to advocate. So results, LA Waterkeeper is really passionate about the LA River, but we know our passion is not enough without support from our community of scientists and our community members. Our goal with the community science program is to engage our community on the LA River and hopefully inspire others to be interested in LA River issues and advocate for positive change. When participants first arrive, we usually take a poll with a show of hands, who has actually heard of the LA River or knows that where we are is the LA River, and there's usually a range. There are usually some folks that do, but there are also folks who weren't familiar with the LA River at all. By the time participants are taking water samples, they've had time to digest an intro to the LA River and its history and challenges, and they're observing the river. As we're waiting for tablets to dissolve and the water quality tests and the PFAS water to drain, this is when they get to ask questions. So we can hear all kinds of things, whether it's what would take to change a value in one of our tests, or how about the studies we reference? Can we tell them more? Sometimes we also get questions about what is happening on and around the river. My favorite question, though, is when folks ask about the best ways to engage with the LA River. What can they do? Because that is what we really want to see as a result of our program. We want our community to be as excited as we are about the LA River. Thank you.
Thank you, Melissa. I'm sorry. Thank you, Maggie. I was notified that we're running a little bit late, so I'm going to cut my comments very short. I want to thank you for your presentation and for LA Waterkeeper's efforts to really educate the public and get the community science involved. I'm a big proponent of and a cheerleader for community science. We use it for our biodiversity program, specifically iNaturalist. So I think that that's great. So thank you very much. Our last speaker is Dr. Melissa Turcotte, Head Environmental Engineering Specialist for the Los Angeles County Department of Public Works. She will be talking about stormwater capture BMPs, effectiveness monitoring. So, Melissa?
Thank you, Mas. My name is Melissa Turcotte. I'm with LA County Public Works. I work in the Stormwater Quality Division. Our purpose for our division is to capture the stormwater for the unincorporated areas of LA County. So this is an aerial photo of the East Los Angeles Sustainable Stormwater Capture Project that has been recently put into the ground, completed. And part of my team's job is to monitor the BMPs and look at what that, how much water is being captured and how well is it doing its job. All right, so this is LA River adjacent. It's not in the river. We're capturing stormwater from the unincorporated county of East Los Angeles. This project was installed in different medians. The medians were underused. And due to the drought and irrigation restrictions, the areas were not doing very well. They were not pretty in any means. So we took 7 medians and we put in a multi-benefit project. This is a rendering of the actual project, what's going on underground as well as above ground. So we took the median itself, we installed DG pathways for walking, bioswales, exercise equipment, things that the community told us that they wanted to see in this area. And underground, we have pre-treatment units, a diversion from the stormwater to capture the water, treat it, and then put it into dry wells. We have 103 dry wells, 4 pre-treatment units, there's bioswales. We have a pilot study with UCLA that incorporates biochar, looks at the effectiveness. So our job was to look at the monitoring of the stormwater capture project. And while we're out there monitoring, let's look at the effectiveness of what has been put into the ground. So we intend for the BMP performance evaluation to help us quantify what we are capturing, analyze any of the system limitations. We've had a couple of different snafus that we've been able to identify using the monitoring equipment, and we can go in quickly and change something out or put a new filter in or change something with the design to then tweak it a little bit. We hope that the monitoring data and the effectiveness evaluation will help us to design future projects, so those tweaks can be pushed forward into second generation, third generation stormwater capture projects. And we're using some of this data, the real-time data that we're capturing, to inform some of the models that we have been using to build these projects in the first place. And also, there are monitoring requirements for many of the grants that we apply and get. So this is the performance index framework. And so the framework was meant to be modular, so it has different categories that we examine. And then, the initial thought was if we can observe all of these different categories separately, then we can, come up with one number. This BMP is X number efficient. We've learned since that looking at each individual category helps us inform what we can improve or how this one category is working effectively. Our 3 categories are water quality, hydrology, and O&M, operations and maintenance. So we will look at the water quality category first. So this is a depiction of one of our stormwater capture BMPs. We have some sort of pre-treatment, the stormwater diversion, pre-treatment. As it exits, it goes into some sort of infiltration chamber or a dry well. So we have strategically placed monitoring equipment at the influent, where the water comes from, and the effluent after it's gone through pre-treatment and before it goes into the ground, so that we can look at the quality of the treatment. So the BMP Performance Index is going to look at how well does the BMP treat the stormwater. So we look at the concentration of the effluent divided by the influent pollution concentration. And if the BMP is performing as we expect that the effluent has less pollutant in it, and therefore the ratio will change. If the BMP is underperforming, there are some cases where the pollutant, we've seen mathematical errors, we've seen other things where the effluent pollution concentration is greater than the influent pollution concentration. There's something wrong with that BMP, so we'll go back in and we'll try to fix it. Here's an example of the system at work. The flow direction is underground in the storm drain. This is an influent manhole where we're going to open it up and take a grab sample. The pre-treatment unit is in the middle, some nutrient separation baffle box where it takes the trash. It also separates the sediment. And then there's an effluent manhole on the other side. Right, our next category is the hydrology category, and this uses some of the design features of the project with the actual data that we're collecting during like a rainfall event. So in this, we're asking the question, how well does the BMP capture stormwater? So we are usually diverting stormwater into a capture system, and it's been designed to some standard. We use the 85th percentile storm design, and we use rain gauges in or around the area to measure the amount of stormwater that has occurred in the rain event. So if the ratio is greater than 1, we have stormwater bypass, meaning the stormwater has filled up the BMP, and now you've got stormwater not going into your actual BMP system. And if your ratio is greater than or less than or equal to 1, the BMP should be performing as expected. So you're in a good area. On the other side, you have actual capture volume divided by design volume capture. So this is the actual amount. We put flow sensors in the influent area so we can see how much water was actually captured. BMPs are considered failing if there's a bypass that's occurring for a small storm. So you expect all the rainwater to go in. If it's a small storm, the BMP should fill up to a certain amount. You're done. If there's higher than anticipated bypass, the BMP should reach the capacity, but it doesn't. So there may be something wrong with your slide gate or something wrong with the design, depending on what the design features are. All right, and the last area we're working on right now is for the operations and maintenance. We've put in these projects. They're supposed to last 30 to 50 years. We want to make sure that we're doing the best we can to operate those BMPs for the lifetime of the project. So that's why these are TBD here. Oh no, this was supposed to be in different slides. I will try to walk you through the slide. Okay, so the blue line in the background is what we would expect from a theoretical performance of your BMP. You start your BMP after construction, it has peak performance. You go through your, your maintenance schedule, so you go back up into peak performance level and back and forth, right? Operations, you observe, maintenance, you fix, you go back up to peak performance. There are a couple of different water quality points that you're looking at, like after a storm or right after your BMP has been constructed. And you have the ability to go out and inspect these things visually between the times that you're going out and sampling for an event. So you have many different points in time, and you can, see what the actual performance curve is. That's kind of where we're going, learning. So we've developed an O&M application, so we're collecting standardized operations and maintenance data, so we can see what points and categories should be optimized for this tool. All right. Thank you so much.